Welcome, dear students. I am Dr. Suraj, Assistant Professor of Botany of Barakpur Rashtor Guru Surendranath College. Today, I am going to discuss about dark reaction of photosynthesis. In my previous class, I have already discussed light reaction of photosynthesis, and today I will discuss dark reaction of photosynthesis. Actually, dark reaction is also known as carbon fixation reaction. So, first of all, I am starting with Kelvin cycle, dark reaction, Kelvin cycle. This is a series of biochemical reaction which reduce carbon dioxide to a 3-carbon sugar. Okay, and it took place in the stroma of chloroplast of eukaryote. And Melvin, Kelvin, and Andy Benson, they actually get Nobel Prize for this reaction. Okay. Now, what happened during Kelvin cycle? You can see here all reactions of Kelvin cycle. Actually, it has 3 phases. First one is carboxylation of CO2, and second was reduction of 3-phosphoglycerate, and third one is regeneration of CO2 acceptor RUBP. RUBP is ribulose bisphosphate. Okay, so as I mentioned, in Kelvin cycle, CO2 acceptor is RUBP, ribulose 1,5 bisphosphate, and in the first reaction, that is in carboxylation reaction, the enzyme Rubisco is involved. Actually, Rubisco enzyme has 2 functions: Rubisco carboxylase and Rubisco act as oxygenase. Here, Rubisco act as carboxylase. Now, what happened actually? CO2 will react with ribulose 1,5 bisphosphate. Which one is CO2 acceptor here? In carboxylation, it will convert into 3-phosphoglycerate. This 3-phosphoglycerate is the first intermediate stable product during Kelvin cycle, and this 3-phosphoglycerate is 3-carbon compound. That's why this Kelvin cycle is also called C3 cycle. Okay. In the next step, which one is reduction step, 3-phosphoglycerate will be converted into glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is a carbohydrate, and this glyceraldehyde 3-phosphate, I am coming later what happened in details. Actually, this glyceraldehyde 3-phosphate will be converted in next sucrose and starch, and some portion of glyceraldehyde 3-phosphate will regenerate ribulose 1,5-bisphosphate again, and this way this cycle will be continued. Now come in details. In carboxylation reaction, you can see here the reaction. What happened? Ribulose 1,5-bisphosphate first will be converted in an intermediate by the enzyme Rubisco carboxylase, and this will immediately convert into 2-carboxy-3-keto D-arabinol 1,5-bisphosphate, and this is also unstable. So immediately this will converted into 2 molecules of 3-phosphoglycerate. As I said, you can see here, this is the first stable intermediate of Kelvin cycle, and it is a 3-carbon compound. 1, 2, 3. It is a 3-carbon compound. That's why Kelvin cycle is also called C3 cycle. Okay. So this is the reaction details. In reduction reaction, what happened? This 3-phosphoglycerate will be converted into glyceraldehyde 3-phosphate, which one is a carbohydrate. Okay, and finally in regeneration, as I already mentioned, that in regeneration some portion of this glyceraldehyde 3-phosphate will again regenerate its own component, that is ribulose 1,5-bisphosphate, and some portion of glyceraldehyde 3-phosphate will produce starch and sucrose. Okay. Now here is the details. Details means how many molecules are involved and how many molecules will be produced. All the details are given in this cycle of Kelvin cycle. So look at very carefully that first of all, you can see here that 3 molecules of ribulose 1,5-bisphosphate will react with 3 molecules of carbon dioxide to produce 6 molecules of 3-phosphoglycerate during carboxylation reaction. Okay. Then during reduction reaction, this 6 molecule of 3-phosphoglycerate will be converted into 6 molecule of glyceraldehyde 3-phosphate. Okay, and you can see here that an intermediate also produced, that is 6 molecule of 1,3-bisphosphoglycerate, but this is unstable. And you can also notice that to do this reaction, 6 molecule of ATP and 6 molecule of NADPH will be required. And now the question may arise, from where this ATP and NADPH come? This ATP and NADPH, as I mentioned in my previous class, that it will come from light reaction. Okay, so 6 molecule of glyceraldehyde 3-phosphate, 1 molecule of glyceraldehyde 3-phosphate will exit this cycle, and later it will be converted into sucrose and starch. So in the cycle, 5 molecules of glyceraldehyde 3-phosphate is present. Among these 5 molecules of glyceraldehyde phosphate, with the help of 3 ATP molecule in regeneration procedure, will be converted again into 3 molecule of ribulose 1,5-bisphosphate. So this cycle will be continued in this way. Okay. In the next part I am coming, there is another CO2 concentration mechanism is also present, which is C4. Here I want to mention you that in C4 plants, a special type of anatomy in leaf is present, which is known as crunch type of anatomy. Why this is crunch type of anatomy? I am coming. Just look here. Normally in C3 plants, chloroplast is present only in mesophyll cell, but in case of C4 plants, chloroplast is present in 2 types of cell. One is mesophyll cell and another one is bundle seed cell. So actually this is one difference between C3 and C4 plants in the anatomy of leaf. Okay. Another thing is, this C4 cycle is also known as Hatch and Slack pathway. Why? You can see here that MD Hatch and CR Slack, they elucidated this. That's why it is also known as Hatch and Slack pathway. Now, come to the reaction. What happened during C4 cycle? Okay, just look at this diagram. Here, atmospheric CO2 acceptor is phosphoenol pyruvate. In C3 cycle, CO2 acceptor was RUBP. In C4, CO2 acceptor is phosphoenol pyruvate. Okay, so atmospheric CO2 acceptor phosphoenol pyruvate will react with CO2, and the first reaction is carboxylation. In carboxylation reaction, it will produce a C4 acid. This C4 acid will be malate or aspartate. Okay. From mesophyll, C4 acid will be transferred into bundle seed cell, where decarboxylation of this C4 acid, that is malate or aspartate, will be happened. So as decarboxylation, that means CO2 will be released from here. This CO2 is start with Kelvin cycle. Okay, so this CO2 will enter into Kelvin cycle. Now, after decarboxylation, a C3 acid, okay, C3 acid will be produced, and this C3 acid will be transformed in mesophyll cell, and here regeneration of phosphoenol pyruvate will be happened. Okay. Actually, these are the enzymes which are involved in the reaction. In first reaction, that is in carboxylation reaction, phosphoenol pyruvate carboxylase will, that means this reaction phosphoenol pyruvate carboxylase is involved in this reaction. In the second reaction, that is NADP malate dehydrogenase will be involved. In third, aspartate aminotransferase will continue this reaction, that is oxaloacetate plus glutamate, that will produce aspartate and alpha ketoglutarate. So you can see here, these are the enzymes which continue this reaction. Now, coming details. What happened? So, look at here. This is phosphoenol pyruvate, and phosphoenol pyruvate, with the help of phosphoenol pyruvate carboxylase enzyme, it will produce oxaloacetate. Okay, oxaloacetate. This oxaloacetate is 1, 2, 3, 4. Oxaloacetate is a 4 carbon compound. That is why this cycle is known as C4 cycle. Okay, so first intermediate of C4 cycle is oxaloacetate, and this oxaloacetate is a 4 carbon compound. Again, malate is also a 4 carbon compound. You can see here that malate dehydrogenase continue this reaction, that is oxaloacetate will be converted into malate, and this malate is a also 4 carbon compound. Here, this 4 carbon compound malate will be transferred into bundle seed cells, as I mentioned earlier. Here, with the help of malic enzyme, decarboxylation reaction will be taken place, and a 3 carbon compound pyruvate, that is 3 carbon acid, will be produced, pyruvate, and this pyruvate then will be transferred into mesophyll cell again, and here it will regenerate phosphoenol pyruvate. So look at here, what happened during this reaction, that is from oxaloacetate to malate. From oxaloacetate to malate, 1 molecule of NADPH will be required. Here also in decarboxylation, again it is decarboxylation, so NADP plus will again converted into NADPH, and also its carbon dioxide will be produced obviously, so this carbon dioxide will enter into the Kelvin cycle. So this is all for C4 cycle, and I want to mention the difference of C3 and C4 cycle here. Actually, in C3 cycle, RUBP was CO2 acceptor. Here PEP, phosphoryl pyruvate, is CO2 acceptor. Also, crunch type anatomy is one difference. In C3 cycle, crunch type of anatomy absent. In C4 cycle, crunch type of anatomy present. There, that means in C3 cycle, first intermediate was a 3-carbon compound, but here in C4 cycle, first intermediate, which one is a C4, that is 4-carbon compound. In C3 cycle, finally, what we saw? We saw that ribulose 1,5-bisphosphate will be regenerated. But here in C4 cycle, what we saw? We saw that phosphoryl pyruvate will be regenerated. Now, come to another mechanism of another carbon dioxide concentrating mechanism. Which one is known as CAM mechanism? That is carboxylation acid metabolism. So, where in desert, during daytime, it is too much hot, and obviously water scarcity for the plants. So these camp plants adopt a special type of features. They open their stomata during night, when temperature is not so hot, that is, temperature is cool. Now, what happened? Oh, I want to mention that some plants, these are the plants, Crassula, Kalanche and Sedum. These are the plants in which CAM pathway is observed. Also pineapple, vanilla, agave, cactus. Actually, desert plant. Now, come to the pathway. Okay. Here, two figures are taken. One figure during night when stomata opened, and one figure during daytime when stomata is closed. So, what happened? During the night when stomata is open, you can see here that CO2, atmospheric CO2 will be accepted here also by PEP, that is phosphoryl pyruvate. When we discuss about C4 cycle, we saw that PEP is CO2 acceptor. In CAM pathway, here also PEP is CO2 acceptor. So, with the help of PEP carboxylase, it will produce oxaloacetate. It will produce oxaloacetate. Then from oxaloacetate, this oxaloacetate will be converted into malate, then malic acids, and you can see here whole the procedure in vacuole. Now, from where this phosphenyl pyruvate came? In chloroplast, by various biochemical mechanisms, actually starch will be converted into phosphenyl pyruvate. Okay, so starch in chloroplast will be converted in phosphenyl pyruvate, and this phosphenyl pyruvate will accept atmospheric CO2, and with the help of phosphenyl pyruvate carboxylase enzyme, it will produce oxaloacetate. Oxaloacetate will convert it into malate, another 4 carbon compound, and ultimately malate to malic acid. Then it is going to vacuole. And what happened during daytime? During daytime, when stomata is closed, what happened? This malic acid from vacuole, this malic acid will be converted into malate, and from malate it will be converted into pyruvate. This reaction is decarboxylation reaction. Malate to pyruvate, this reaction is decarboxylation reaction, and it will produce CO2, and that CO2 will enter Kelvin cycle. Ultimately, what we see that ultimately all the plants ultimately follows Kelvin cycle. But how? With the help of this procedures. So, okay, now malate with the help of malic enzyme will be converted into pyruvate. This pyruvate will be converted again some various biochemical procedures to starch, and starch will be stored here in chloroplast. So both these procedures needed for plant for their survival. Okay, now come to the last part of this topic. Okay, so photo expression. Actually, Otto Warburg observed that oxygen inhibits photosynthesis in C3 plants. Okay, oxygen inhibits photosynthesis in C3 plants. This phenomenon primarily it was known as Warburg effect. So you can say photo expression is also a Warburg effect. I have already mentioned to you during Kelvin cycle that Rubisco has two dual roles. First one is Rubisco carboxylase and second one is Rubisco oxygenase. In photorespiration, oxygenase activity of Rubisco will be observed. Now, actually, what happened? At low CO2 concentration, Rubisco performs oxygenase activity when low CO2 concentration, but when it is high, it will act as carboxylase activity. Okay, so low CO2 concentration, Rubisco will perform its oxygenase activity. Oxygenation of Rubisco leads to the production of one molecule of 3-phosphoglycerate and one of 2-phosphoglycerate. Now what happened in this reaction? I am coming. Okay. First of all, I want to mention that photorespiration requires 3 cellular organelles. First one is chloroplast, second one peroxisome, and third one is mitochondria. So photorespiration require 3 cellular organelles: chloroplast, peroxisome, and mitochondria. Now, what happened? You can see here that ribulose 1,5-bisphosphate with the oxygenase activity of Rubisco, it will produce 2-phosphoglycolate, and this 2-phosphoglycolate will be converted into glycolate with the help of water. So, and this glycolate will be transported in the peroxisome, and here you can see that this glycolate will be converted into glyoxylate. Okay, glycolate will be converted into glyoxylate. Here 2 molecules of H2O2 will be produced. Remember that in previous CO2 concentration mechanism, that means C3, C4 or CAM plant, neither will produce H2O2. But so, as I mentioned, that in photorespiration 2 molecules of H2O2 will be produced. In the next step, this glyoxylate will be converted into an amino acid glycine. In previous CO2 concentration mechanism, no amino acid is produced, but in photorespiration, glycine, one amino acid glycine is produced. Next, this glycine will be transported into mitochondria, and in mitochondria, glycine will be converted into serine. And look at here, one molecule of ammonia will also produce here. Okay, one molecule of ammonia will also produce here. Now serine will transported into peroxisome from mitochondria, and in peroxisome, serine will be converted into hydroxypyruvate. Serine will be converted into hydroxypyruvate. In the next step, hydroxypyruvate will be converted into glycerate, and this glycerate will be transported into chloroplast, and here glycerate will be converted into 3-phosphoglycerate, and this 3-phosphoglycerate will enter into the Kelvin cycle. So ultimately, it will also follow Kelvin cycle. So, in photorespiration, what we saw three cellular organisms is required: chloroplast, peroxisome, and mitochondria. It is a 10-stepped procedure. You can see here, as I already discussed. In this procedure, we observe that hydrogen peroxide, that is H2O2, is produced. Amino acid glycine, serine will be produced, and ammonia is also will produced. Okay, so come here. All the 10 steps which require these enzymes. In the first step, ribulose 1,5-bisphosphate oxygenase activity. In the second, phosphoglycolate phosphatase. In the third step, glycolate oxidase. In the fourth step, catalase enzyme. In fifth step, glyoxylate glutamate aminotransferase. And in sixth, glycine decarboxylase. In seventh step, serine hydroxymethyltransferase. In eighth, serine aminotransferase. In ninth step, hydroxypyruvate reductase. And in 10th, glycerate kinase. So these are all enzymes which carry out photorespiration. So with that, I am ending my lecture, that is dark reaction of photosynthesis. Thank you.